Okay, I want to welcome everyone to today's session. It has been one step at a time, and it has also been so great and awesome. I I hope we have been doing something with what we are learning. I hope that we have been putting them into practice. So during our last session, we concluded the toxicological screening, where we are able to screen our data for toxicity. We laid that foundation that our major concern is the safety. Safety is our major concern. Before we even talk about the therapeutic properties, our major role there is safety. So which we have taken our time to screen our data, and we're able to select the ones that are safe. For tonight's training, I'll be merging two modules together. I'll be combining the drug likeness module and the natural product mining module together. So I will combine both of them together, and that will end, because we have been discussing about plants. If you watch from Phytoinformatics, it was about the plant. To chemoinformatics, it was about our plants. Down to toxicological screening, it was still about our plant. So we are really paying much attention to this plant, because we must make sure that we have a standard from our plant. Then whatever the plant can do, maybe in the aspect of the physiological activity, that will not really be a problem. But the problem is, let us ensure that the plants we are using in building our formulations are safe. That is why most of our attention has been given to our plants, But then we shall end that aspect by focusing today on how we can screen our natural product also. These are plants, how we can screen them to pick the ones that are drug-like. You know, like I always tell us that the reason why we always ask for 1,000 and more compounds is so that at the end we can filter off the ones that are not necessary. So there are two conditions now that we have been able to set. Number one, when you have your 1,000 and more compounds, you first of all screen them for toxicity, which we have taken our time to cover that aspect. You screen them for toxicity. Then the ones that passed the toxicity test, remember the essence why we say screen for toxicity, so you can compare the ones that passed and the ones that failed according to the number of compound you have. So it will give you an idea that, see, this my plant, a good number of them are, a good number of the bioactive compounds are toxic. So you begin to, it will now shaping you to know the necessary step you have to take to reduce that toxicity. But if a good number, a good percentage of the bioactive compounds are safe, it's giving you joy that this plant I'm using will definitely not be toxic. So that is the first set of screening that we have done. Now, the second set is we are going to screen also to see if we have drug-like compound. If we have drug-like compound. So we are supposed to only focus on the ones that passed the toxic screening, then pick those ones to screen them for drug likeness and lead screening, lead and drug likeness screening. But because we are not after developing, we are not after lead, like we are not after doing isolation and synthesis at the end. We are after formulating this plant. So we are going to use the whole data we have, that same data, we use the whole data also for our lead screening and drug likeness screening. But if your idea was to achieve a potential compound, then you're going to focus on those ones that passed the toxicity screening. Then you shall now pick those ones and subject those ones for drug likeness screening. Well, our idea of this training is to see how we can come up with herbal formulation. So we still focus on the whole bioactive compounds too. So at the end, we shall also see out of 1,000 compounds, see the number that are drug-like. So it's giving us idea of the potential of this plant. So that is what we have to do tonight. So let me quickly share my screen as we begin. Once again, thank you for coming around tonight. We really appreciate your effort. Right, so can you see my screen?
Yes, sir.
Yes, sir.
Right. So we shall be looking at drug likeness models and lead screening of natural products. So I'm combining the two models together because they have similarities. So we'll just combine them so that when next we meet next week, we have two more sessions. So those two sessions will now cover up for the bioinformatics and molecular docking. So we just have 3 modules left after today's. We have the bioinformatics, the pharmacodynamics, and pharmacokinetics. I might even push both of them together as pharmacology, because both the pharmacodynamics and the pharmacokinetics, they are branches of pharmacology. So we'll see how we can wrap them off by next week. So a quick one. Drug likeness models and lead screening of natural products. Now, I will just wrap up this introduction aspect, then we move into house training. If we're able to finish on time tonight, then I will introduce bioinformatics. Now, because of the chemical diversity, and the biological relevance of natural products often present challenges in drug development. So let's take note of this. The chemical diversity, because you see that your plant has a diverse chemicals. So because of that diversity and the diversity also in biological relevance, these natural products often present challenges in drug development due to issues related to their molecular complexity. So this challenge often arises from their molecular complexity, poor solubility, and low bioavailability and potential toxicity. One of my students yesterday or on Wednesday shared a research done by some postdoctoral students. What they did was they were working on biopesticide, just similar to what we are doing in Hendeb. But they noticed that biopesticide, they want to spray it as an aerosol. So they want to make it, you can spray it. So they noticed that particular natural product they got from the plant was insoluble. So they had to look for a way to make it soluble. So these are challenges you may have in natural products. They are complex their molecules are too complex. Some of them are soluble, some are non-soluble. Some have good bio availability, some have low bio availability, and we cannot tell. We can't tell which is which in that plant, but we can now screen the plants to see the potential. To what extent? To what extent is the bioactive compounds in this plant feasible for this my study? To what extent are they available? Because whatever that plant can do is as a result of the bioactives, the bioactive compounds in that plant. So if we can assess the bioactive compounds, do this screening, and be able to tell that this plant has a lot of compounds that have low solubility. So it will guide you in your dosage formulation. We might now screen those bioactive compounds, and then we'll see that these compounds in this plant to have their structures are too large. So can they be absorbed by the bloodstream? So this will guide you in your formulation. Now, consequently, drug likeness evaluation and pre-docking lead screening have become indispensable. So you can't do without drug likeness and pre-docking lead screening. All these, most of us usually skip it. If you even look at some journals, some literatures, once they are brought in their lead down and they are protein, they start doing docking, which is wrong. You have to screen your data for lead. We call it pre-docking lead screening. You have to check if this product has the potentials that could make them a drug. So I'll not waste my time docking these compounds. Immediately I can modify it, because we are not just after the therapeutic effects, but we are after the safety and ability of these compounds to act as a drug. So this part is totally indispensable. And it is an early stage strategy to rationally prioritize promising natural compounds before engaging in computational or experimental studies. So with the idea we shall be sharing today, you can now prioritize your natural compounds before you can engage in your further computational studies or experimental evaluations. That overview of drug likeness. Overview of drug likeness. Drug likeness refers to the qualitative and quantitative assessments. So we are assessing both the quality, that is the character, and we are quantifying them numerically. Because if you must be a girl, then there are certain qualities you must possess that will make you a girl. So those qualities, we are going to now convert them to numbers numerically. So it will assess us to what extent can you be a girl? Can you look like a girl? So if we must get a drug, whether it's a natural or synthetic, the formulation we want to make, we must assess to what extent is the bioactive compounds in that plant, to what extent do they look like drug? Because drug has a model that for you to be a drug, there are certain criteria you must possess, you must pass before you can become a drug. So those criteria are called drug likeness. Criteria that makes you look like a drug, because you're not yet a drug, but they will make you look like a drug. So when these characters are not found in you, so you're not drug-like. Now you can imagine a plant, and this plant you have 10 bioactive compounds. And out of these 10 bioactive compounds, based on in silico evaluation, 8 are not drug-like. 8 of these bioactive compounds, the plant has 10 bioactive compounds, and 8 out of these bioactive compounds are not drug-like. That is to tell you that even if you proceed in making a formulation from this plant, it will not sell, it will not be effective because they are not drug-like. So all this training is to make you make wise decisions while you are building your formulation, because at the end, we are looking forward to see our participants, maybe as group, as colleagues, coming up together and said we are embarking on a formulation. It will not even be a crime. It won't be a crime that if at the end, because it's not about the training and we train you, then you go. It will not be a crime if at the end we form a team, or everybody here form a team, and we say, okay, we want to come up with a drug with a herbal formulation. We all put our heads together, because the only place where we start having issues is in the lab, wet lab validation. We start looking for bioactive, start looking for reagents that will be needed in the quantification. So drug likeness is very necessary before you go to your docking. It helps us to assess whether a chemical compound possesses the physical, chemical, and structural properties that are consistent with known drugs. So to every known and approved drug, we have seen that there are some, we notice some consistent parameters that we have found in all the known drugs. So we have to highlight those consistent properties that we have found in all known drugs. We now come to conclusion that for you to be a drug, all these drugs, they have something in common based on their properties. They have something in common, and that is to say that for you to be a drug, you must also have these properties in common. That if you don't have these properties in common, you may not pass the drug development stage. So that is what we are going to look at today. And if you look at it, you may not have a software that can do all what you want, but you can now leverage on data analysis to develop functions that will help you do whatever you want to do at ease. Now, drug likeness models does not guarantee, very important, it does not guarantee biological activity, but it indicates the likelihood that a compound can be developed into a safe and effective therapeutic agent. So here, it is not telling you that the compound is effective against malaria or is effective as anti-cancer. No. But our concern is, if at all that this compound will be effective, can it be developed into a safe and effective drug? That is what we are looking at. So can these compounds in my plant, can I formulate them into a drug? So that is the whole overview of drug likeness model. So we shall look at these models. I will give you 5 models. There are basically 5 common drug likeness models which we subject our compounds to our data to. One of the simple, most common is the Lipinski rule of 5. So the Lipinski rule of 5. Lipinski rule of 5 is the most widely used drug likeness model. Now, it has been designed to predict. Now, even if we are always using the word prediction, because we cannot use the word like accurately, so we use prediction. But I still want to emphasize that this prediction, they are highly precise. It's just like what we did in Excel. You write a function, and the function reads that these compounds are toxic. You begin to question yourself. This function I've written, to what extent is the function true? To what extent is that formula valid? That is just what it is. Because we had group 17 toxic parameters. We wrote a function, and the function reads that this compound is toxic and not toxic. Now you begin to question, you now begin to argue that function. To what extent is this output from my Excel based on the function? To what extent is it true or valid? Don't you think it's just a mere prediction? But this is the challenge we usually have when we are teaching in silico. People get biased. We had a training 2 days ago, which will conclude on Saturday, tomorrow, on the beginner's course. So yesterday, while we are introducing computational chemistry, one of the participants asked a question because we generated some chemical data. So they asked to what extent is this data valid? Because we really need to rely on them. You need to tell yourself the truth that these data I'm generating are reliable. So the participant asked that question. So what I did was we drew a compound in ChemDraw, generated some chemical identities, some chemical properties. Among that chemical properties were the molar mass of that compound. So the molar mass given to us was 78.05 based on the system, based on the software we are using. Now, and I said, okay, this is computer prediction. Let us manually calculate the molar mass of that compound by ourselves, based on how we are taught in chemistry. So we did a manual calculation. It gave us 78. And what is in the system was 78.05. So I threw back the question to the person that asked that question. I said, now, to what extent do you trust this data? The person was saying, but there is 0.5 at the end of 78. I told him that the one in the computer is even more accurate because it was working with the atomic mass units, the isotopic mass. But we are calculating with approximated mass. So that is to tell you that whatever this system is giving you, it's not just giving you something that it feels like. It's giving you precise data. So why I'm explaining this is so that we learn how to build confidence in our in silico data. Then we can now go to the lab to validate what we have gotten. Not to redo it again, or is now to validate this result. Okay, I've seen this result, so let me just back it up with the lab work. Because wherever nobody will accept your in silico result as a final result. They would like to see a proof they will tell you from the main experiment, from the main experiment we know. So that is where we are, how we have found ourselves now. So one of the models is Lipinski. It is designed to predict oral bioavailability based on simple physical and chemical properties. And on my screen at the right, you can see those properties that we are used to design that model. So if you look at the right, you see those properties. Now, they have our scientists have taken their time, and they have seen that there are some properties that play a role in oral bioavailability. That is for a drug to be taken orally. So for maybe we're now developing a herbal product in tablet form, which will be taken, which will be swallowed. So to what extent does the bioactive compound there contribute in the solubility, in the bioavailability of my formulation? So we have some parameters that the molar weight of your compounds should not be, they said, should be less than or equal to 500. The log P, which is for lipophilicity, the extent of that compound to dissolve or to be soluble in fats, to be soluble in non-aqueous media or environment, that the value should be less than or equal to 5, so not more than 5. Not more than 5. The hydrogen bond donors of that compound in your formulation should not be not more than 5, so less than and equal to 5. Then less than or equal to 5. Then the hydrogen bond acceptor should be less than or equal to 10. So that means from 0 to 10 should be less than or equal to 10, so not more than 10. So these parameters now contribute to the Lipinski rule. So what's the implication is that compounds within this space, within these regions, are more likely to be orally absorbed. Now, violation correlates with poor permeability or solubility. So if a compound fails these parameters, this range given to us is telling us that those compounds may not be soluble. Now, it also implies that excessive violation increases attrition during development. So you find it difficult in developing effective formulation from those compounds that failed this, that violated these conditions. So that means that we shall be using these conditions and their range to screen our compounds, to screen our bioactive compounds. The next rule, the next model is the Weber or GSK rule. It's saying that the Weber model emphasizes on the molecular flexibility and polarity. So here is after the polarity, which strongly influence oral bioavailability. So at my right, we are seeing the parameter, just 2 parameters here, and they are acceptable range, the TPSA and the rotatable hydrogen bonds, the rotatable bonds, so that the TPSA of that of those compounds that must be in your formulation should not be more than 140 Armstrong, 140 square Armstrong. So less than equal to 140. So if it is more than 140, some will say at most 150. So if you can meet this condition, so imagine that you have a good number of bioactive compounds meeting these conditions is giving you a green flag. Or when you have most of them violating these conditions, don't know what will be your opinion, what will not be your decision, it's just a red flag. So this guides us at the early stage of herbal product development. The implication is that low TPSA improves membrane permeability, because it's talking about the surface area. So low TPSA, if your TPSA is very low, it helps to improve the penetration, membrane permeability. So it helps to improve the penetration of that compound. Limited flexibility improves binding efficiency. That is from the rotatable bonds. You the flexibility, how you can rotate, whether rigid or flexible. So if you have limited flexibility, it will improve binding efficiency. So if your rotatable bonds is low, not more than 10, then it's telling us that such a compound could likely bind very well to your receptor. Strong is also a strong predictor of oral bioavailability. So it also contributes to oral bioavailability. So we shall include these parameters in our screening. We shall include the parameters in our screening. All right. So we shall go ahead to the third model, that is the Gauss filter. The Gauss filter, What is it all about? The DOS model defines draw likeness using physicochemical and structural descriptors, including molecular size and polarizability. So at my right, you can see the parameters established by Gauss filter, that the molecular weight is between 160 to 480, the log P, which is T for solubility, for minus 0.4. So all these are structural properties. I really don't know, but people who did this are amazing. That from your structure, they can generate properties, just like how we can calculate the molar mass from structure. That is how they were able to calculate these parameters, the value from the structure. So they were able to calculate it from the structure, just like how we can look at a structure and we say, this structure is not soluble in water. Like if you look at sodium chloride, you can easily tell that sodium chloride cannot dissolve in ethanol. And then you can now estimate to what extent will sodium chloride not dissolve in ethanol. But if you look at water, you see that water will be soluble in ethanol. So if you look at your sugar, you say that your sugar will be soluble in ethanol, but your salt will not. So that is how they were able to calculate these values we are using. So feel free. The challenge is always the acceptance. Because I did a survey a long time ago on the knowledge, perception, and attitudes of early career scientists towards data generated from computational simulation. The result shows that most people usually say, this is computer work. Somebody laid a complaint to me that his professor has refused to approve his research work because it involves computation, that all this on a computer generated figures. So it is a big challenge to us. So that's why I'm just trying my best to give you a little explanation that will make you to some extent rely on this data. So the Gauss filter has some implications that it ensures optimal molecular size and steric balance. So here it looks like it always look at the steric effects, the electronic effects. The electronic effects, that is what Gauss filter was based on. Now it filters out extremely small or bulky molecules. So when your molecule is too small or too large, then the Gauss filter can help us to filter it off. It is useful for lead-like compound identification. So you can now identify lead compounds from there because those compounds are not too small, not too large, so they fit in appropriately. They fit in properly to the conditions for a lead compound. So you can see the molar refractivity, the total atoms. So these conditions too are what makes up the Gauss filter. Then the hidden rule. Some of these rules are not found in ADMET, but most of them are found in the Swiss ADMET. So you can see them in Swiss ADMET. The hidden rule. The hidden model focuses on intestinal absorption, integrating lipophilicity and polarity. It has two parameters.
Change your slide, sir.
You say?
You change your slide, and you have done that, sir.
No, I am on hidden.
We've not done it.
That's what I'm saying.
Just,
you don't remember to change the slide.
Okay, you're not seeing the hidden?
We can see it now, sir.
Okay, all right. Thank
It's
you.
okay now.
Okay. So the hidden rule mostly talks about the intestinal absorption, integrating lipophilicity. There is an idea that came to my mind while I was preparing for these lectures in the afternoon. I think I will share it with us. Let me not destroy the class. So we'll discuss about it if we have time tonight. Because there are a lot. There are a lot to consider if you want to develop an effective herbal product. There are a lot to consider. Too many things to consider. So the Eden rule that focuses on the intestinal absorption. So there are two conditions for it: the Lord P, which is T for solubility, and the TPSA. So here TPSA is 131 Armstrong square or squared Armstrong. So the Lord P is no more than 5.88. Why Lord P for the Lipinski was not more than 5. So the impression is that it can predict human intestinal absorption. So today, each and every one of us shall have our model. Like today now, we shall establish a Hendeb model, while you too you establish your own model. So you can see people just choose some parameters and develop a model for it. Give it their name. So you can choose some parameters like we do tonight and develop our own model. It helps to provide simple and effective. It's simple and effective for oral drug candidates. Okay. It complements the verbal rule. So these are the implications of the Eden rule. And then the last one, which is the model or the bio rule. The model rule. The Mughal rule. I noticed that the Mughal rule has a lot of parameters that make up the model. But what is it talking about? This model integrates multiple descriptors. It integrates multiple descriptors to define drug-like chemical space. So the model integrates so many descriptors to define drug-like chemical space for lead optimization. The impression is that it is suitable for diverse chemical libraries. So the model is very useful when you have natural product that has a diverse bioactive compounds. It's a good balance between strictness and flexibility. Highly relevant to natural product screening. So we shall also deploy model rule in our natural product screening. So these 5 are the basic drug-like models that you can subject your natural product to and screen them. But what we shall be doing tonight is we will not use any one, like each one. We'll combine the whole parameters for each of these models, then use it to build our own model. So we combine, because all of them are all necessary. So we shall use them to now build our own model. So you can call it your name. But we shall be building, for me, I will be building Hendeb model. So each time I have natural products, after I have screened them for their toxicity, I will now screen them for their drug-like properties using Hendeb model. Now, there is another parameter called QED. Now, QED is not a model, but QED is a continuous scoring model. Like it's not a drug-like model, but it's a scoring model. Like, let me use the word for ranking. It's a ranking model. It's a ranking model that's good. It's excellent for ranking. It's a ranking model that you can use to rank these bioactive compounds that we are successful in either the Lipinski, the Ghost, the Weber, or the Ida. So when you have some bioactive compounds that you have subjected them to drug-like models and they were successful, you can now rank, sort, filter, or arrange these compounds to the ones that have better priority, better likeness priority. So you can use the quantitative estimate of drug likeness to now rank these bioactive compounds. Like you may have students whom we are assessed, and those students were brilliant. So they were assessed for their intelligence. So, and after the assessment, 10 students were found to be brilliant. So they termed these 10 students brilliant. Then you may now want to rank these 10 students that has undergone several tests and has been proven to be intelligent. So you may now want to rank them according to their level of intelligence. So that is where QED comes in. So when you have compounds that have succeeded the drug likeness models, you may now decide to rank them using QED. So, and it has range. So the ranking will now may even tell us, oh, that among these 10 students that we are termed brilliant, we have the ones that are extremely brilliant, the ones that are brilliant, and the ones that we are moderately brilliant. So from the QD range, anything 0.5 and above is godlike, just godlike. 0.7 and above is highly godlike. Less than 0.5 is moderately. It's totally just moderate. It has a moderate potential, but yet they were termed as drug-like molecules. So you can now use QED. QED is found in ADMET lab has it, but I'm not sure if your Swiss ADMET has it. But ADMET lab has the QED, so you can use it and do your own screening. So it's excellent for ranking of compounds. It reduces bias of rigid rules. It's highly recommended for natural products, and that is why we are here. So quickly, we are going to move into hands-on. This is a summary of the drug-like properties and their range. So I was able to summarize the properties that defines whether a compound will be drug-like or not. I arranged them with their minimum value and their maximum value. That is their range. So we have how many parameters we shall subject them tonight? 1, 2, 3, 4, 5, 6, 7, 8, 9. So there are 9 parameters here. These parameters we are cumulative from the drug-like models. So we shall use them to screen our compounds at the same time. What we're doing tonight may answer a question that I don't know who raised that question. If we have parameters that their score are not unique, like somebody asked, maybe you want to screen both molar weight that has 100, 200, and you want to screen maybe your toxicity value that has 0.1. How do you screen these things at the same time? So I think that tonight's class may also give a better answer to that question that was raised during our last session. So do we have questions before we move to the hands-on tonight?
Yes, I have a question, sir.
All right.
Looking at the summary of the drug-like properties and their range, we can see that for hydrogen bond donors, we have minimum value of 0, maximum value of 5. Then for hydrogen bond acceptors, minimum value of 0, maximum value of 10. Does that mean that, the question now is this, does that mean that we can have a drug-like compound that will not have any hydrogen bond donors at all? I will still be drug-like. Because
Okay,
according to what you taught us in pharmacophoric properties,
yes,
you told us that at least hydrogen bond donor must be there, at least 3.
yes, I get that range. Yes.
Now that the value is now giving us 0 here, is this one now telling us that a compound can be drug-like without having any hydrogen bond?
All right.
I hope
Now
my
that
question
is
is clear.
simply, yes, it's very clear. That is simply what the model gives. That's 0 to 5. So it's a very technical question. Doesn't that imply that I can be drug-like without having hydrogen bond acceptor, and I can also be drug-like without having any hydrogen bond donor? Yes, but I usually give my value between 3 to 5, because from my own experience, this is a model that was built, a model based on Lipinski. So, but from my own experience, what I usually do is, since we are told by our fathers who were in this field before we came in fact, we are told by the people who handed over to us. So since we are told that hydrogen bond should be between 0 to 5, also why not I now build compounds that, since they say no more than 5, but I won't accept 0. So I prefer taking from 3 to 5. Why for the hydrogen bond acceptors, I prefer taking 5 to 10. Reason because from experience, I have seen that that there must be hydrogen bond donor and there must be hydrogen bond acceptor. And these two, yes, and these two usually take part in one of the interaction called the conventional hydrogen bond interaction, and is a vital interaction in a receptor in drug interaction. Is that hydrogen bond, conventional hydrogen bond interaction is vital. So for it to be there, you must have this presence of hydrogen bond acceptor. So I don't take that 0. So while we'll be building the model now, we'll take 5. The one that is 0 to 5, we'll take 3 to 5. And one that is 0 to 10, we'll take 5 to 10. Then the rotatable bonds, 0 to 10, I will take 5 to 10. Then the number of rings, 0 to 7, I might take 2 to 7. So because I don't accept 0, because those things must be there. Their presence have role to play. So thank you very much for that wonderful insight.
You're welcome, sir. Thank you, sir.
You're welcome. Okay. Do you have any other question? All right. So we work with like I said, you may decide to work with the ones that passed, the ones that only pass your toxicity screening. But that idea is, if you are interested in developing a lead compound, like the interest is to identify a potential drug candidate that will be further synthesized or isolated. But if since we have a collective goal here of herbal formulation, so we have seen here, to what extent we are our compound toxic. Even self, I forgot to add percentage, although we calculated it that day, but let me just do the calculation. We are saying this value, so this value divided by 17, then times 100. So, all right. So it stripped me my mind to do that day. All right. So this will enable us see to what extent, to see the percentage. Then we can easily rely on the percentage. So now, since we have equally seen to what extent can we rely on these values based on this data, based on their toxicity, we can also see to what extent should we rely on the bioactive compounds based on their drug-like properties. So what we'll do is, I think we still have that data. We had a raw data, yes. So that we will not stress ourselves going back again. So I'm going to make sure I have those 9 parameters here. Remember, this was the raw data we downloaded from ADMET, but ADMET may not have all the parameters we are looking for. So let's use Swiss ADMET this time around, or I could better just check. So let's just check one by one if ADMET has all those parameters. I need to minimize my screen so that I can be able to see the parameters, because I may not carry them by heart. Right, I'm coming.
Sir, please, I have a question.
Yes, go ahead.
Yes, you divided that value with 17 and multiplied by 100. Why? How did you get the 17?
Okay, I took them to percentage. During our
Yes, when you were there.
During our last session, we had 17 toxic parameters.
Okay.
So why those values? Okay, those values we are the one that passed the 17. So we look at the percentage out of 17. All right. So let's see if our original data, so it will not stress us again. We have the molar weight. Let's see we have the TPSA. We have the number of hydrogen bonds. So I will just indicate the ones we have by red, so that we will not go back again to start downloading. So we have this one. This one is among. Hydrogen bond donor is among. TPSA is among. Number of rotatable hydrogens is among. Number of ring is among. Yes. So which other one again? Okay. Number of. Okay. I've taken log P. Let me check for log P. Okay, my log P is among, so I can see my log P. Okay. I don't think if ADMET has carbon atom, but we can neglect the carbon atom. What is remaining now is molar refractivity. Molar refractivity. Let's be able to know what ADMET used to represent molar refractivity. I don't want to stress myself going back to ADMET. Boiling point, melting point. Just to know what is molar effectivity in ADMET. Okay, I will just have to do. I just have to go back to ADMET, so I can easily get the molar effectivity from there. So I will know the acronym for molar effectivity, the abbreviation that was used. Okay, so but for the purpose of the training, we can still do without it. Then later on your own, you can identify the molar effectivity. That'll be okay. Is that okay by us?
Let's just get the molar refractivity now, since you're already
Okay, sir.
We should look for it.
let's look for it.
Hello, I want to know your
What
opinion.
happened?
Should we do away with it then? Maybe your free time, you can identify which of the parameters here is actually for the molar refractivity. Okay. I'm already there, Sha, so let me just get it. All right. So let me just copy one and paste. So all right. Molar refractivity. Supposed to be under physicochemical properties. So if we can't get it here, it means that will come in from Swiss ADMET. Okay, you can get that one from Swiss ADMET. Okay, it's not found in ADMET. It's not found in ADMET. So we'll do without it for the sake of the training, but I know you can get you can pick it from Swiss ADMET and combine with your own, because I'm sure it must be here. Okay, so this is the molar effectivity 168.69. So you can get those whole data, those whole smart code, paste them in your paste them here and copy that smart code and copy the molar effectivity to make up your data. So we may not be going to that extent now due to time, because at least we are just being exposed to what is involved. So let us do with what we have now. We have, if I have to count, we have 1, 2, 3, 4, 5, 6, 7. So 7 out of 9, because I took off carbon atoms, not necessary. Then the molar reactivity, since this data we are from ADMET, so we'll just focus on these ones. But what you do, maybe when you copy the whole smart code now to your Swiss ADMET, then download the result. I should better just do it once and for all, so that every one of us can see, to avoid doing your own projects. So once I copy this whole smart code, then I'm going to the Swiss ADMET. So I paste the whole smart code and I run. It's 20, so let's see how fast it will be. You are submitting molecules that are 2. What are you telling me? Oh, I don't know what is happening now, but it seems we'll just continue because. Okay. Let's see. Let's expect a limit of 200 characters per smile. Don't understand. I don't understand what. Don't understand what is happening to smiles now. Okay, once I just do this once now and this last one, and it's not responding, I will just leave it.
I think one of your compounds is too large. I've experienced it using Swiss ADMET before. If it's too large, it will not read it.
Well, my dear, please, let's, instead of 1 parameters to hinder us, let's just continue with the 9 we have, right?
Sorry, I feel one of
So
your
what do we do?
compounds there is too large. That's why you're not.
What
That's
do we
why
do? This
Swiss
is what we're
ADMET
going to
is not
do.
doing it.
We are going to copy out the parameters we are using. So let's do it together now. Take this my code, hold your control, I like your molar weight. I like I may not spend that time to start sorting, so we can just do it with the bulk. Report that one compound is more than 200 characters. So, but it will to start identifying that compound and begin to, that should be the first compound. The first compound is about 1,000 this. But the essence I'm trying to say is that we can actually live activity in your own personal time, include it to your data. The most important is to see how we are building the parameters, the screening. So even though you add the molar reactivity, you can still build it. You can still build it. So highlight your smiles, highlight the molar weights. I'm already holding my control, so now to do that, highlight from the number of hydrogen bond acceptor to number of ring. Then go to, I will come back later for QD, but go to Lord P now. Highlight your Lord P. All right, so after you have done all that, you copy Ctrl C. So can we do that together? Are we together?
Yes, we are.
Yes, sir,
Hello?
we are. Yes, sir, we are.
All right. So having copied it, you come to a new worksheet and you paste in a new worksheet. So these are the 1, 2, 3, 4, 5, 6, 7. So we want to use these 7 parameters for our screening. We want to use them to build our model. So we want to build our model from these 7 parameters. Now, if you look at them, their ranges differ so much, but that will not stop us from building the model. It's a very simple task. What we are going to do now, so the first thing you will do is to we are going to copy this range. We'll copy this range, these parameters, give a space, and paste here. So the first thing we'll do is that we'll convert this their value into binary, into a binary of 1 or 0. We'll convert all these values into a binary of 1 or 0. That is what we shall do. And so that is why we copied. So why I copied these parameters to another column is to enable us convert each of these values we have here. We'll convert them into a binary digit of 1 or 0. So that will also help us to do our screening when they are now at 1 or 0. So how do we do that? We agree that if this molar weight falls within the range of 150 to 600, let it say 1. We were using 1. But if it does not fall within the range, let it say 0. So are we together? That is what we are doing. We write a function that we read our molar weight and we tell us if this value is between 150 to 600 based on the range for molar weight. If it is within that range, then it will tell us yes by saying 1. But if it is not within that range, then it will say 0. So how do you write the function? Molar weight is in your B2. That is B2. So we'll come to this molar weight. Let me zoom my screen so that we can see it properly. So you come to the molar weights. I'm actually keeping this Excel so that my slides, so we can be seeing the range. That will enable us in. All right. So let's come here and say equal to. This is the function you write. You say equal to. You say if. After if, you open bracket. You say and. If and, open bracket, B2. if and open bracket B2. So just like the way we used to write, we say not below 150. That means we say less than or equal to 150. Because we are saying not below 150. So if B, okay, we say if B is greater, because the minimum is 150. That means it must be greater than 150 and less than 600. So it must be from 150 and above. So we'll say if B2 is greater than and equal to 150, comma. Also, if B2 is also, now it will be now less than, if B2 is less than and equal to 600. So that is the range. If B2 is less than and equal to 600, you close the bracket, you say comma. So if the value is within this range, let us say 1. But if it's not within this range, say 0. Then you close bracket and enter. So it's telling us 0, which means that the molar weight found in this B2, 1,699, did not fall within the range of 150 to 600. So we can drag it down to see others. So are we together?
Yes, sir. Yes, sir.
I didn't get the
All right. We just converted.
so the
We are together,
You didn't
sir,
get what?
but it's
The
not clear.
function?
yes function.
Okay. No, we can't
Right
be
from the
together
time
when
you
it's not
copy,
clear.
sir. Right from the time you copy.
Oh. What I copied was the data we had already. Remember, we had those data already.
Exactly.
So I just went to those data, the one we downloaded from ADMET. So I took the parameters that defines draw likeness properties, copy them to a new sheet. Let me even rename the sheet. I can rename the sheet.
So together with this mic,
Together
with this Michael.
Yes.
Together with this Michael.
Because this SMILE code will help us to identify. Yes, with the SMILE code. Because the SMILE code is what we use to now identify those compounds. So having copied the data, having copied the data to a new sheet, the next step we'll do is to convert those values into binary digits of 0 and 1, so that all of them will be uniform. Because if you look at some of them, we cannot use the same function we used last week because they are not, their ranges differs. So we must bring them to one range, which is 0 and 1. So what we are doing is write a function that will check that value, the value for molar weight. Read it if it falls between 150 to 600. Now, this function we are writing with Excel here is also the same way they use in building the software that does this thing for us. So, and if now see that some of those softwares you pay to access them, the ones that are good enough, you pay to access them. So, but in our sense of paying, we are developing a means where we can use our data analysis tools to do them for us, and we can even rely on the results very well. So the function said equal to, you say equal to, when you write equal to, you say if is for the condition, open bracket, and means that it will be within this range. After and, you open bracket, molar weight is in B2. So if B2, since we are saying between 150 to 600, that means not less than 150. So it should be equal to 150 and above. So we say if B2 is greater and equal to 150, also if B2 is now less than or equal to 600, that is the range we just built now. Equal to, greater than or equal to 150, then less than or equal to 600. If that be the case, you close brackets. Let the system tell us 1 if it is between. Let it read 1. But if it's not between that range, let it read 0. Then you close your bracket. You press enter. So that now this 0 here now implies that the value, that molar weight, is not within this range. So you can drag it down for yourself to do others. So it is not really difficult. Once you write the right function, once you have the right function, you can do others. Then you come back to this. To me, what I will just do is I can highlight down, but if you highlight down, you must change their range. So what you do now is to change the range, because I just highlighted what I did for molar weight for others, but the range for hydrogen bond acceptor is not the same thing for molar weight. The range for hydrogen bond donor is not the same, so change the range. So let's change the range. Hydrogen bond acceptor is between what? We are saying 3 to 5.
But
So
acceptor.
3 to 5. Yes.
To
What is
10.
there is 0 to 5 now.
0 to
But
10. No.
we'll build it using, okay, acceptor
Acceptor
0 to 10.
0 to 10. 0 to 10.
So let's build 5 to 10, right? Let's build 5 to 10. So we can say 5 here, 5 here, and then here we say 10. We say, so I'm just editing it, and then enter. You scroll down. Haba. This 0 means that hydrogen bond acceptor is right. It's 46. So I really love what we are doing tonight. Or do you think we should maintain the 0 to 10? Well, it's not ideal. There must be hydrogen bond acceptor. Then hydrogen bond donor. Hydrogen bond donor 0 to 5. I can say 2 to 5. So not less than equal to 2 and not more than 5. So I can say 2 to 5. So I edit it 2 to 5. Then you can drag down. uh,
Small. I'm not trying to.
okay. It's not easy. TPSA. TPSA is saying 20 to 150. So you edit it, edit the range. Twenty to 150. So it's just easy. You edit, then you drag down. Okay, it's interesting. Yeah, now the number of rotatable bonds 0 to 10. So I can build it at 2 to 10. So I'm building my model. I'm building my Hendeb model. So number of rotatable bond 0 to 10. I can say 2 to 10. 0 to 10. So my model, I'm building my model for 2 to 10. 2 to 10. Number of rings 0 to 7. I can say 2 to 7. 0 to 7. I can say 2 to 7. So I can say 2 to 7. Then the last one, Lord P, minus 2 to 5.88. So minus 2 to 5.88. 5.88. Okay, so I'm done with the first stage. I'm waiting for you. Are you done? Hello? Are we quiet? Hello?
Yeah, with you, sir.
All
We are
right.
together.
All right. So I can proceed to the next stage.
Yes, sir.
Yes, sir.
All right. So in the next stage, we will now calculate our score, our lightness score. So let's calculate the lightness score here. And calculate our lightness score. So let me change it to black. Now, so the lightness score will be, remember, the one that gave us output range of 1, meaning within the range, while output range of 0 is out of range. That means for a compound that passed this, we have a six parameters now. So for a compound that will pass these 6 parameters, we are expecting a score of 6. A score of 6. So if anyone have a score of 6, that means that compound was able to, in fact, is hyper drug light because it perfectly fits into the drug light parameters. So we shall do summation, say equal to summation for molar weight to Lord P. So let's see the score. This is score of 1. You drag it down. And wow, I can see score of 6.
Please show the function again, sir. The function.
Okay. summation. Summation, your J2 to P2, you close bracket and enter. So we are summing the binary digits, our 1 and 0. Our interest now is on the one that reads 1. So those one that reads 1 means that they are within the range. So the one that reads 1 is within the range. So we'll now calculate for each of them. Each of them, how many parameters did they pass? How many parameters did they succeeded? Did they pass in each of them? That is what we are doing now. So now the same thing we did now, you can take it to percentage also, since we are expecting so you can read it as a scroll like this, or you can read it in percentage. So you can sort. You can sort it, let it be arranged according to their score. Let it be arranged according to their score. So we can do that sorting. Let it be arranged according to their score. Okay. And we can go to data and we say sort. And we are sorting the likeness score from largest to smallest. It's not responding. Likeness score. What is happening to it?
Didn't highlight.
No, I must not even highlight. I mustn't highlight. I noticed that it's trying to sort and later goes back. So once
go back to home, select, highlight the likeness score, and you will see on the list on the top there, I don't know what they call it. You go back to home, click on your home icon there. Yes. You will see, where is it again? I can't find it anymore. Oh, sorry, I can't find it anymore. Where you can see sort from smallest to largest,
yes.
smallest.
Yeah, it's in data.
It's in data.
Yeah, I've done. We have been doing that, but I don't know why this one doesn't want to continue with current selection. I'm sorting the likeness score. Likeness score. I want to highlight everything, so it will just sort everything. I think I know the reason. Let me see. I think okay.
It's good you highlight everything.
There is a bridge. I think it's due to this space I left here. It's due to that space I left there. So let me see if I can highlight like this. Let me highlight like this. Because the sorting will help us a bit to easily interpret it. Okay. Largest to smallest. All right. Okay, so now we are, as you know, research is always based on the researcher's view. It's always based on the researcher. So now that we are expecting a score of 6. What should be your benchmark? So we are saying 70%. So we always say 70%. So let's take it to percentage.
Sorry.
Let's take it to percentage so that we can look at the percentage clearly. Since we always say 70%, this overseas times 100. All right, so if you are going by this, it means that just these three compounds, we are able to meet up with our drug likeness model. So what is it trying to tell you? That out of 20 compounds, only 3 are drug-like according to the drug-likeness models. Do you wish to continue with the plant, or you may wish to opt out for another plant? Is that your choice?
Okay.
So,
It's okay now.
eh?
I will continue.
so yeah, it means that actually I have drug-like compounds. So in as much as you have drug-like compounds, it simply implies that even though others may not be absorbed easily or other factors, but I have compounds that are drug-like. So you now pray to God. You now have a prayer to make that let these compounds that are drug-like, let them be the lead compound after my docking process. Because even though they're not the lead compounds, but in as much as from your docking, they were able to possess therapeutic properties, they are still good to go. Because we always say that having high binding score is not enough. It's just a hint. Binding score is just a hint. It's just a hint. But let these compounds that pass this criteria, let them be among the ones with a good binding score. If they will be during your docking process, that means you are about developing a good product. You are developing a good product. So at this point, I would like to take questions and interactions. Let's ponder over it. Then you can do your QED. You can still bring in that QED, but right now we have already sorted. So I can rearrange, I can resort the result. I bring them back. Then bring in our QED to see if the ones with the highest score, we agree with our QED. With the highest score in your QED, because the QED is ranking those bioactive compounds according to their degree of likeliness. And our own likeliness now is showing that this first compound with 6 has 100% likeliness. So if your interest now is in developing a lead compound, I think you pay attention to this compound. And let me even take a look at that compound from PubChem to know who is that man. So if you have questions, you can do it to ask questions. I can probably introduce bioinformatics in the next 20 minutes, or what do you think?
It's okay.
So that percentage function, please, can you restate it?
We can introduce them.
Hello, sir, can you hear me?
Okay, I will.
Hello.
Yeah, I can hear you.
Can you restate that percentage function?
Yes, yes. Okay, okay. I'll do that now. So this is the compound that this man said doesn't have a name. So this is the man. Okay, this is the IOPAT name. The man that is giving us that good result. It was created 2017. It's still a new compound. Still a new compound.
So
But if
how
you
do
look
we
at this
do it? How
compound,
do we do it? Just one single name. Because all this
it has a,
1, 2, 3.
it means that maybe for now there is no common name for it. Yes. Because look at the date it was published, 2017. How many years ago? It's still novel. It's still novel. And if you even look at that compound, I don't think if much work has been done on this compound. But to me, the structure is not even showing as if it's something that could be therapeutic. But okay. Well, let me. So if you look at see the content is scanty. There is no biochemical, no medical reports. So I think, to me, if any of you can focus on this compound, it's novel. I see it as novel compound. I see it as something that is still novel. Okay, but we can even check the synthetic accessibility. You can do some things, do more findings from there. So you can do findings of that compound. It shows a promising compound.
Sorry, scroll down.
Let's
Let's
see
see
if
the synthetic ability.
okay. I don't think if PubChem does not show the synthetic accessibility.
So
Let's do
ADMET,
that
let's take
in
ADMET.
Swiss ADMET.
Okay.
Let me see ADMET. Swiss ADMET can give me the synthetic accessibility. Uh, 5.54. 5.54. For 1 is easy, to 10 is difficult. So as the static accessibility ranges from 5 to for 1 to 10. So it's 5.5 for average. Let's see what ADMET we also say concerning
Sorry,
that.
where's your cursor?
Okay, look at it. The last parameter here. Can you see it?
Yes, I can see it.
5.54. So let me see what ADMET will say about it. Let's see what ADMET will say about it. Let's see.
Very hard.
No. It's not it. It's not it. Though. I've not.
Okay.
I've not. This will push us now to concentrate in this compound. But I don't think I'm ready for that now. Oh, it's still very hard. It's hard.
It's hard.
But being hard doesn't mean not possible now.
Maybe we can optimize.
Yes, we can. We can
Can we
optimize.
check the
So
those that have 5?
it now depends on you. It depends on you. It depends on you. Do you want to work? Those that have 5, yes. Those that have 5 what? 5 score. Okay, is that should check for this two that has 5?
Yes.
Okay, let me check. Let me check. Let me quickly take both of them to Swiss admin. So it has been a nice finding tonight, and it's a good one. So you make sure you name this model you have built according to your name. That is your model. So your model now is more comprehensive. If you check other model, their own is not comprehensive, but you just built a comprehensive model now. So you can tell us what your model describes, because each model has what they describe. So what does your own model now describe? So you can tell what your model describes. Okay.
Sir, what does
Good.
describe, sir?
Huh?
What does this
You said what?
your model describe, sir? What does it describe, sir?
My model is my model is a complex model. It describes everything that all the model describes. So my model describes both bioavailability, permeability, solubility, and it's a comprehensive
Almighty
model.
model. Almighty model.
Yeah. Is that my so look at the compound, those two that has 5. 7.09 for synthetic accessibility for this one,
Very
and the other
active.
one is 5.10.
The first one is better. So we choose the first one.
Yes. So
Please, sir, what is the range for synthetic accessibility? The normal range?
from 1 to 10. From 1 to 10, the lower the easier, the higher the harder.
Thank you, sir.
Yeah. Okay. Questions and interaction has taken, this is 9 minutes to 9. Right. So even if we may not take up the bioinformatics now, but what we need in the bioinformatics is not really, I'll just
To 10.
from that to, yes, but give me more 15 minutes. I just
Ask
give
you
me
a question. Oh,
yes. Okay, somebody asked about that percentage, right?
about it now.
Somebody asked me about the percentage.
That's
Is it
the last thing they problem. Sorting under drug line, like let's go. How you are
please,
able to
can you
navigate?
can the person repeat the question, please?
The function for the what's it called, the percentage stuff.
Okay. This is the function. We said the lightness score over 6, or in bracket times 100. The lightness score over 6, because over 6 is our total lightness score. So each of the lightness score divided by 6 in bracket, then times 100.
Okay, sir.
Okay.
Sir, that lightness score, is it like in you just decide to choose a range of 1 to 6? So I don't know, how do you come about that likeness score?
Because the Hendeb model has 6 parameters. Our Hendeb model had 6 parameters, and so each of those parameters should give us a high input of 1. So that means I'm expecting to have a total input of 6, because I have 6 parameters. So if my compound happens to succeed in all of them, I'm coming.
So I thought it's
I
7
can
parameters,
see
sir.
7 parameters.
Wait.
It's 7.
1, 2, 3, 4, 5, 6. Oh, 7. Sorry. So I have to change. Sorry. Oh, see where we are busy praising that number, that first one. So sorry, let me change it to 7. Oh, I just noticed that now. All right, thank you very much. So that means it's still those on a range of 70% above. All right. So since I have
Sorry,
a model,
sorry, the likeness score something. Did you later highlighted everything, or you just click on that column, please?
no, okay, just click on the column, on the first column. Then it will understand for others. So I just, the likeness score is
I
at
didn't get it.
column. Okay, let me do that again. Let me remove this. So let's do that together. So we got the likeness to be 6 already. So now the percentage is equal to my likeness score. Okay, I
No,
open a
not the
bracket.
percentage. It's
Oh,
about ranking from 6 downward.
okay. No, after it ranked, after it's calculated, we sorted to arrange it in descending order.
My question is that did you highlighted everything from molecular weight to likeness, or you just click on that column alone?
No, I highlighted everything from SMICE. From this SMICE down to likeness. Everything from the SMICE down to the likeness. Because I want it to sort in line with the SMICE. So arrange, copy, highlight from this SMICE down to the likeness score. Okay. So the person that asked about my likeness, so I just choose that Hendeb should have a score of 7. And maybe you can even state it, you can even make it a law that for you to pass Hendeb, that Hendeb likeness states that a drug-like molecule must possess 70% of the likeness score. So I just stated that one now as my likeness model. And
So how
for you
do
to
we
have
get the
70%,
cost?
you must not evaluate.
Citation. So if you choose Hendeb, so how do we get the citation?
The citation. Okay.
Yeah, I'll be now.
So I didn't want it to just based on each of the model. I just want to have a cumulative properties. Because if you can have compound that will pass those criteria, you have gotten a good compound. So some of you might call your own Vitor's model. I don't know all your names. Professor Excellence model, Professor Adiola's model, Professor Professor Vincent model. So you use it to do your own, because I don't want to rely. If you look at some of the model, they only based on 2. So, but I want a cumulative model. So since I have 7 parameters, I'm expecting this 7 to give me output of 1, 1. That is if you pass that particular parameter, it should give me 1. So since you want to pass, so if you must pass the 7 parameters, I'm expecting a score of 7. But I can say, okay, that out of 7, if you fail, you must not fail more than 2. So you can also make it as your own. You must not fail more than 2. At least let me be a bit not rigid. You must not fail more than 2. So if a compound fails more than 2, we throw you out. Just like Lipinski. Lipinski, you must not fail more than 1. You must not fail more than 1. If you fail 2, they will throw you off. If you fail 1, it's okay. And the rule has been acceptable. So maybe I will just write I'll write a publication now on Hendeb's model, dual lightness model. So that's by the way. I really want to appreciate us for coming out. So everything about plants, we are done with it. Making sure our plant is fit. So what you now do now is try to correlate between the screening, between the toxic ones. See if the ones that we are less toxic, we are among the ones that we are drug-like. So try to see that correlation. Mostly if your target is in synthesis and isolation. So try to see if there is a correlation there. Like this first one for toxicity. This first one that we accept. So we accept only 70% above. So this first one is see, it's my code. This my code is, so you can trace this my code to see if it's among the one that passed the lightness model. So you can do that cumulation. So it's always if your interest is if your interest is on isolation or synthesis, then you will not use all your data for likeness model. You only use the one that passed the toxicity screening. So those ones will be the one that you subject into likeness model, because you're only focusing on the ones that are not toxic. But here we are still using the whole plant. So our focus is the percentage, the potential of toxic and non-toxic, and the potentials of drug-like and non-drug-like. So if toxic, if the one that we are non-toxic is higher than toxic, you just be looking for a way to maneuver it. Then if your drug-like is more than non-drug-like, you should be celebrating. It means that if that formulation, that herbal product will be taken orally, it will be well absorbed by the body. To be well absorbed, because you have compounds that are absorbable. So it is just giving you idea. Right, I would like to take little question, then we wrap it up for today. So I will combine, like I said, let me see if I can get that module. Okay. All right. So I will see a way of combining these last two, the pharmacodynamics and the pharmacokinetics. There will not be a problem to us. It won't be a problem to us. Why the bioinformatics, we may not bioinformatics is very deep, so we'll just pick the part that concerns us. The part that concerns us. Yeah, we'll pick the part that concerns us. I will see a way of rounding everything off by next week. Then please, I'm pleading that I believe that anything we do now, you are doing it to your data, right? Hope that is what we are doing. Are we doing the same thing to our data?
Trying to. We'll do it.
Hello?
We'll do it.
Hey.
Trying.
No, not. We are trying to now, like I expect. I don't know. I expect maybe. Well, try your best to do what we are doing with your data. The reason is that it will help you to easily achieve your manuscript, your project at the end. So you won't wait till when the training is over, then you now start thinking of what will I do in my project work? What will I do in my research? You start over. So it might be better as we are doing with our data, we are doing same thing with your own data. So once we drop the pen, you're also dropping your pen. So I would like us to know how many weeks can we schedule for the project defense after our own class? Because we have a lot on our table, so we have to plan it. How many weeks can we give you? You know, our mini project that we do is always 2 weeks. For mini project, for our free training, we give 2 weeks after the training. So for this one now, I want you to express yourself so that we plan our activity. Let it not clash with other activities. Because we're almost done. We are done. What is next thing now is the docking, which all of us know docking. Everybody. I don't think there's anybody who have not done docking before, but
Docking, no, I'm new. I don't know anything.
there's no problem. I will still take my time to do that. So how many weeks should we give you? Because the training ends next week Friday. So how many weeks should we give you after next week for the project defense of this work?
Then when we are done, we will conclude that now.
No, we are done already now. After next week, we are done. I just want to know.
That Friday.
Okay, that Friday. Okay. There's no problem. So
I think a week after the last class, a week or 2 weeks after the last class, can schedule the defense.
Somebody will hit you.
Wow, 1 week.
A week, wow.
Stand
not
on.
Mini project. This is main project. Main project, not mini project too. A week to be too small. Project.
2 months.
Question concerning the project.
2 months.
Yes.
Yeah. In what's it called? Preliminary assignment on the phytoinformatics. When we collated plants and the chemicals that we downloaded them in the SDF format, and those SDF formats are stored in a file as separate SDS formats.
Yes.
By the time we are putting them into OpenBabel for conversion into smiles, the mode of transfer in terms of import from that folder is going to be different from the one we did when we downloaded derivatives from the PubChem. So what I want to ask now is that by the time we are loading into Babel, will it take it from the folder directly?
Yes. Yes.
Okay. All right.
Maybe if you have 200 compounds in the folder,
yes,
and you can copy the Control A. You can highlight the 200 compounds at the same time and open.
Okay.
We did, that was what we did.
Okay. Yeah. I get it. That's okay. That's okay. I understand.
Yes.
That. Thank you. Yeah.
All right. So, why I call on those that have a question? Let's use this weekend and design your topic, then drop your topics to the platform. Or will it be better? Yes. Yes, just design a topic you are working on, and then let's drop the topic to the platform so we can document it. And look, we'll give 1 month. We'll give 1 month after the end of this training, because some persons are taking their MS exam, some are taking their PhD exams and other stuffs. Some that reach out to me, their systems, some of them the laptop is having issue. So let's give that grace of 1 month. I think a full 30 days is enough. If we are giving mini project 2 weeks, so let's give this project, although this one should not even take time because we are already doing it in situ. Yes, we are already doing it in line with the training, so it should have taken time, but we'll give 1 month. We'll give 1 month so you can prepare your manuscripts. Like I said, our plan is at the end, let's see if we can publish your manuscript. The only thing we may do is to see if we include MD, but even if MD is not there, it will fly because it's a herbal formulation we are doing. So the topic might, I want to do, but know that we are aiming towards herbal formulation, polyherbal mixture. Yes, polyherbal mixture, but we are just using 1 plant. But you can just design the topic how you want to design it, and let's see it probably this weekend or before the next training. So we see what we are doing. I just want to get prepared before we finish, so I can face the other training. Those who have registered for green chemistry, we are kicking off on the February 10th. 10th of February, we're starting green chemistry. And we have released a new training that is Bioinformatics. It's a 2 days, it's like we do now. We know already in Hendeb that in every month, the first week of that month, we have free workshop. The second week is our webinar. The third week, another free workshop. And the fourth week is a beginners training. So the beginners training we had this week was awesome. So this whole thing continues every month. Although some persons complain that we should be giving them space to breathe small. Let them breathe small. But I don't think if we will grant that request. So we just want every month, we have a lot packed every month. So Bioinformatics, because we introduced Bioinformatics to us as part of the module for this training. But the idea now for that course by first week of February is every participant will build a database. Even if it's not the one that we are going to publish, we'll learn how do they create this database. This Bioinformatics database we are sourcing for, how can we create our own? How can we create our own database? So that is what we are focusing on, building a personalized database. So join us. That training is free. Okay, I will take 2 hands up here. I'm okay. And Abiodun. Victor, do you have a question or we have already answered the question?
No, that's the question. You talk about likeness being 6. Say 6 is okay. What if all of them are good? Like all of them are 1, and they
That
are
means
7 because 7
it's perfect. That is perfect. Very, very good. You should be celebrating. It's as if you're in heaven.
I found out one of my own is 7.
Very good. Very perfect. Your plant have drug-like compounds. So you should be in heaven by now. It's very good. All right. Abiodun.
Yes,
Professor
sir.
Abiodun.
Thank you, sir. Thank
Yeah,
you,
thank
sir.
you.
So I just want to do a kind of a recap from the phytoinformatics so down
Okay.
to chemoinformatics. Like for
Mm.
instance, now after retrieving the phytochemicals, the bioactive compounds, so it should be followed by toxicological screening, just to know those that are toxic and those that are safe before we further
Yes.
into the next step.
Yes.
Are you getting me now?
Yes. Yes. Yes.
So now move down to the remaining parts like drug
Yes.
likeness properties
Yeah. And then
based on the
okay.
one we had today. Then after
Yes.
getting the let me say the lead compounds, we now
Yes. We
move
now
down
do
to
okay.
the pharmacokinetics and pharmacodynamics for the docking
Yes.
process, just to back
Yes.
everything up.
Yes. Yes.
Oh, all right, sir. Thank you, sir.
Mm. All right. So once again, I appreciate you for being around. It's not easy to keep it up consistently. Some persons here followed has been on a training all the week. In fact, almost every day. Some persons here join us in the free training. Okay, like this week now, we had our own training this Tuesday, Wednesday, and Thursday. Some persons here join us in that training. And now Friday now, we are here again. Saturday, tomorrow we are rounding up the beginner's course. You still come back again. It is not easy. So I really want to appreciate you, especially all of you, for your efforts. Even we must appreciate you because it is not an easy work. It's not an easy work. Even though it is not easy on our side, but at least the little token we are taking from you is helping us to buy some juice after now to refresh our system, then come back again. But for you, it is really difficult. I know when we are learning some of this years ago, it is difficult, and I really want to appreciate you. Appreciate you so much. Yeah. We are looking.
I think you also deserve a lot of appreciation, because I, for one, I have never been interested in herbal products, and my cold attitude to researching herbal products was based on the manner in which I saw people do research in herbal products. It wasn't intellectually tasking at all. And for me, it wasn't appealing to me until I started, using some bioinformatic tools about some years ago to do my research in pharmacology. And that was actually what opened my eye into, the possible use of in silico, which for me is not just intellectually tasking. It is also, interesting because the way and manner people are doing herbal product research, it was more like, very crude and very hidden. There is there is no light into what they are doing. And for me, I don't like anything that I cannot explain, But when we started using these computational tools, I found it more interesting. But the challenge that somebody like me faced was finding a platform like this, to interact, to learn new skills and all that. And you can correct me if I'm wrong. I doubt if there is anywhere in terms of our higher institutions where this kind of knowledge is packaged in such a coordinated manner, the way you are packaging it. And I'm advanced in my career in academics and research. But despite my advancements in academic and research, I find this very interesting, and I'm happy I'm learning new things, on your platform. And apart from that, I'm also very encouraged by the quality of your audience, especially the young ones. Majority of the people that I said are doing research in herbal medication that makes it much interesting to us are also advanced people. But unfortunately, they didn't have the opportunity to get this kind of platform to enrich their skill, to do research the way they are supposed to be doing it. But here we are seeing very young people, and that is my greatest joy, that the young people you are impacting this knowledge today on, by the time they also advance in knowledge, it will be a very big boost to our research in Nigeria across board. So you don't know you cannot yourself estimate the gravity of the impact that you are, establishing on Nigerians' research capacity, now until maybe about five years later. Then when you begin to see the fruits, then you will know that you are doing a very great job. So we feel a sense of what's it called, indebtedness to you. The token that we are paying you does not equate to the amount of job and the good job that you are doing, not just for individuals, but for our research, society in Nigeria. So kudos to you, and I really appreciate you. Thank you very much.
Thank you. I just felt that I'm now in heaven. When you feel that you are in heaven and I should not come back again. So, but I have to come back. Thank you so much, Olabi, with me for that. Your name sounds like my brother's name, Rotimi in River State. So I don't know if you are connected to River State.
We are not
but
no, I'm not connected to Rivers in any way. I'm from Ogun State, but it's a popular name anyway.
Yes. So thank you so much. I appreciate that encouragement.
Dr. Erisa.
Yes, sir.
I'm sorry. When we are discussing the rules, you wanted to say something that you want to discuss with us. You now said later after the. I don't know whether you still remember.
Thanks to I was trying to talk about
you said
that there are so many conditions that you must consider in terms of herbal product formulation. Okay, fine. That is it. It will be a long time discussion, but let me just give you a hint. Do you think, okay, the human body is an aqueous system. The human body is an aqueous system.
Yes.
Aqueous system. Now,
yes.
That means that if you are formulating drugs that will work, if you are formulating drugs that will work in the human body, I'm just sharing the idea. It just came, but we have to refine it. Now, if you are formulating a drug that will work in the human body, so if I take a plant extract, if I take a whole, maybe a powder, a tableted powder, if I swallow the powder, now this powder has so many compounds But because the human body is an aqueous system, I'm thinking that it should be those bioactive compounds that are water soluble. Those ones are the ones that will exert their physiological effects on the body. Because if they are not absorbed by the body, you cannot do anything. So why the ones that were not absorbed will be excreted? So now that means if I am designing a herbal product, so I should focus on those compounds that are likely to be absorbed by the body. And that means those compounds must be water loving, must be water soluble. Except designing a compound that will go to the brain. That means if the compound is going to the brain, that compound must be lipophilic. Because the brain is so, but outside the brain, I'm looking at for compounds that are aqueous in nature. So in your extraction, because now you have done a formulation. I want to characterize, want to extract and characterize. So we should focus more on aqueous extracts. We should focus more on aqueous extracts. So it is those aqueous extracts probably that will be the ones that will exert the physiological effect in the body.
Those are hydrophilic, right?
Yes. So we can actually think about it, and probably we can drop our opinions in the platform.
It's true, but I'm also trying to consider from the structure of the cell. All these things, they work at molecular or cellular level in the body system.
Yes.
And from the phospholipid bilayer of the cell, they have a kind of hydrophilic head and hydrophobic tail. And those tail, they are folded inward. So for a drug to pass through the cell membrane into the cytoplasm, it has to be lipid loving as well. So those that are water loving, they'll be dissolved at the head, the surface of the cell membrane. So we have to consider that one as well. The lipophilicity and hydrophobicity. Those that are those that are lipid soluble and those that are water soluble.
That means
so we have to
we must
consider the two
have both
factors
of
as
them.
well. Yes, we have to consider them. But the one will now be more than the other based on the site of action of the drug in the body system.
Yes. So that means our solvent is still based on what we have said, polar and non-polar in your extraction.
That's why the essence of log P.
Yes. Yeah. That is that is just the useless idea that came to my head this afternoon. So
well, if I may contribute to that discussion, you see, the absorption of drugs, especially orally, depends a lot on a number of factors. Yes, the hydrophobicity and the hydrophilicity are part of it by virtue of the nature of the cell membrane. But apart from that, if you look at those criteria, there are other parameters that are also very important, especially the size. And bearing in mind that the process of permeating the membrane, that is the process of absorption itself, also depends on the molecule. There are some molecules that have specialized transporters that they don't even need, that their hydrophobicity or hydrophilicity. The transporter just picks it from outside and sits inside. So all of these are all of these are just models that sort of guide us in terms of when we are trying to develop our compound. By the time they pass this first hurdle, right? Okay, we are not there yet, but again, it's like you are taking, we want to admit people into the university. You first do your jump. When you pass your jump, then you go and do your post jump. So this is stepwise. This is the first hurdle that you're going to have to overcome. If you overcome that hurdle, then you go through the next hurdle. It doesn't mean that if you pass jam, you'll be admitted. Exactly. So and again, and that's why they've combined so many factors terms of
Exactly.
the log P, which is the water
Factors
obtain
together.
what's it called, partition coefficient, the size of the molecule, of the
Yeah,
hydrogen
yes,
bond,
yes.
as the reflecting bond,
Yes, that's a,
that
yes,
is
yes. The
the
molecular weight.
molecular weight and all that. So all of these are taken
Yes.
into consideration, considering all the processes that are involved in drug absorption, especially through the mouth. But again, another question I want to ask, which is also coming out of that, does this apply? I mean, I'm not, as I said, I'm not a herbal person. Is this applicable to one, biologists?
Yes.
And even drugs that are not going to be administered orally, let's say parenteral drugs. I don't know if anybody can give me a clue on that.
Yes, yes. I did much on that under membrane biochemistry as well. Yes, yes, yes.
Okay, I think I'll look at the webinar we have for February. I will look at the webinar we have, and I have to change the topic to this discussion. So yes, that's yes. If I check the webinar we have for February and the topic is not interesting, I will change it to this, so we can look at the molecular level of drug.
All right. All right, sir. All right, sir.
I appreciate.
Thank you, sir.
Maybe
Yes,
maybe yes.
Hello?
Yes,
Hello?
I can hear you.
Am I
Yes,
on?
yes, we can hear you.
Yes, sir. Yes, sir.
Thank you. I want to question on the issue of lipophilicity and hydrophobicity. Majorly, what actually contributes to the toxicity of the drug is if the drug is highly lipophilic. So that's why they are actually guiding against drug being highly lipophilic. So that because being lipophilic will actually enter into the cell and stay for long. So that is why majorly, that is why when you look at the drug, the phases of drug metabolism, there is what we call phase 1 and phase 2. All of them is to render the drug more water soluble for the purpose of excretion. So the drug has to be more of hydrophilic more than the being lipophilic.
There are lots. There are lots. Like I said,
yes.
I will just have to change the discussion we have for this February second week webinar to this, and probably most of us will be part of the speakers. So if I reach out to you, just be ready to deliver that job. Thank you so much. Thank you. I appreciate the great intellectuals we are having here. Please, after this training, we are not leaving. No, we are together. Yeah, we are together. All right.
There must be a strong communication that must come.
Very strong one. Very strong one. Yes. Very strong one. Oh. Very strong one.
Actually by you, so it's actually complete it because they say that job of everybody is job of nobody.
Yes. Professor Justina, do you want to say something?
Good evening, sir, and others. Yes. Hello.
Yeah,
so
I can hear you.
I want to add that I have the same observations, perceptions that my colleague Rotimi was expressing. I want to add that before I joined the first training, which was free, I didn't have any interest in researches on plants. I was doing some other things. But for obvious reasons, when I saw, I don't know, something just caught my attention and I saw that training and I felt like I should attend. I did everything I could and I was admitted and I struggled through. Then came this one. Remember at the end of the other one, I said that I was interested in the toxicity studies. So as we started this one and we got to the toxicity test or state last that was last Friday, last Tuesday. I want to mention that I have derived a lot of benefits, educational benefits from these trainings. On Tuesday, we had a postgraduate seminar in my department, and I would have been completely lost by the presentations of one or two students who had to do with all of these herbal formulations, docking, this and that. Yesterday, we had an inaugural lecture, and it was so much on what we have done in the first training and so far, and I was easily carried along. So I want to thank you thank Hendeb Training Center, and you, our teacher in particular, for having us have this level of exposure. And I thank you for your patience. Thank you for every of the ways you're handling things and us. It's very encouraging. And I believe that we are going to derive more benefits, even for our future researches and teachings. And I'm very grateful. I'm asking that God will help you to continue to help our younger ones by this opportunity you are providing. Thank you so very much.
Amen. Thank you. Thank you. Oh, I'm so much grateful. Thank you. Okay, we have to take a bow at this point and say a very good night to each and every one of us. Thank you so much. In as much as God keeps us alive, we'll keep doing what we know how to do. And in as much as I'm also teaching, I don't joke with training. I also attend trainings. We are also improving ourselves while we teach the ones we have learned. So we keep learning also. There are some training I'm also paying also to learn. And as I learn, I look at it, I improve, I add my own knowledge, and we release it to the public. So once again, thank you so much. We appreciate you. Good night, everyone. Thank you.
Yeah, good night, everybody.
Yeah.